Good afternoon, everyone. Welcome. It's exciting to see everyone for our first Science Cafe of 2024. Happy New Year. Hope the new year is treating you well. It's been an exciting one here at the MDI Biological Laboratory already. So we're looking forward to a great season, another season of our Science Cafes, another wonderful series of opportunities to engage with all of you and share a little bit about some of the exciting science that's happening not only at the MDI Biological Lab, but around the world. And that really brings us to our speaker this evening. We are very excited to have Dr. Anastasia Paulmann with us. She is currently a postdoc in the laboratory of Dr. Hermann Haller, who is also with us this evening. And she is currently a student at Hannover Medical School in Hannover, Germany. And she's, as I mentioned, doing her postdoctoral research fellowship with us here in Salisbury Cove. So we are really, really excited to have you with us, Anastasia. Welcome.
Yeah, thank you very much, Sherry.
Welcome also to Dr. Haller, who's going to join us as well.
Happy New Year.
Wonderful. So Anastasia, I know, a big part of the work that you are doing here at the MDI Biological Lab this year is really developing, in many ways, a new model to help us understand aging, and in particular, aging of the vascular system, which is interesting and something we don't always think too much about, but we're excited. I know you're going to talk a little bit more about the details of your work with us, but before we do that, I thought maybe we'd like to just get to know a little bit more about you and what brought you to MDIBL, kind of how you became interested in the work that you're doing. And I know Dr. Haller had a big part in that, right? And so I thought, Dr. Haller, if you might just kick us off by talking a little bit about your mentoring, why you as a nephrologist feel it's important to have, students like Anastasia in your lab or fellows like Anastasia in your lab, and what benefit does that bring to a physician? Why would you, want to do research like the kind that Anastasia is doing as a nephrologist?
Yeah, I'm happy to do that. In fact, Anastasia, hopefully you have to ask her why she wants to do that, but I can answer the question in more general terms. We have MD PhDs in Germany, but the educational process is different in the two countries. So in Germany, you start in medical school and you first train, and that's the basis for becoming an MD PhD as a fellow. And the fellowship is 5 years. In most cases, it's more like 6 years. And then you're a full-blown internist and nephrologist. And during that time, you are doing mostly clinics. You have been doing a thesis. You have been working in the lab a little bit, most of them during their studies, actually, before they started their fellowship. And Anastasia has been working on endothelial cells, endothelial cell culturing, questions of endothelial cell differentiation. But then she became an excellent medical doctor. She's one of the best doctors in the department. And then a lot of fellows actually stay in clinical medicine. But she kept her interest in research and basic research, and then she embarked on something, imagine, which is another 3 years at least for doing basic research. So this means in Germany, an MD PhD is 8 years in training, and then you have an understanding of clinical medicine, but also of the mechanisms of disease. And then you go back, and this is the basis for an academic career in Germany. Anastasia, when she will go back, that's the plan at the moment, she will start her own lab, she will build her own lab group, and she will become a senior nephrologist in one of the university hospitals in Germany, and then start an independent career. And then we can see her in a couple of years as an independent department chief in Germany and running a large research group. So this is what she is doing at the moment. But now she has just started. She has started on a very adventurous journey because it takes a lot of guts. You are already an established clinical nephrologist. I mean, people consider you to be experienced. You do biopsies, you do ultrasound, you counsel younger doctors and younger fellows, and then you start at MDI in a lab, and you start with methods, and you start with a new animal model. First, you try not to make your patient survive, but to make killifish survive and understand the aging process of killifish. This is where we are at the moment. And just one more interesting fact is that all the German postdocs within the US system, and there are quite a few of them. At Harvard, there are more than 200 at the moment. They are supported by the German government for a 3-year, 4-year period in a foreign country to learn and to study, and then come back and build the core of the German MD PhD program. So that's the background. This is what Anastasia is doing now. So all of a sudden, after being an educated fellow, she is now a junior researcher at MDI Bio.
That's great. So Anastasia, as Dr. Haller mentioned, you didn't exactly pick the easy path here, right? So you decided to try to create a new research model, which has in and of itself presented some challenges, I think, for you. But I'm wondering if you can start by just telling us a little bit about this research model, how you see it really having the ability to give us some insights into how humans age, because it doesn't really initially, we don't necessarily think about studying fish to help us understand how human disease, the human disease process. So I'm excited to hear a little bit about that from you, and then we can talk a little bit more about some of the specifics of your work. But if you want to just jump in, that would be great.
Yeah, thank you very much again for having me. So my name is Anastasia Paulmann, and I'm a medical doctor from the Hannover University of Medicine. And I've started my work here at the MDI BL now about half a year ago. And I had the great opportunity to start basically not a new model, because the fish I'm working with is maybe not the fish you're all familiar with, the zebrafish, but I'm working in the African Turquoise Killifish. So if you hear me say the word ATK, this is our very short, lovely abbreviation for our fish, so we can say it very quickly. So the ATK is a new animal model which we're trying to develop as a canonical aging model. And the good thing about this fish that it basically does the aging work for us. So we don't have to do anything with this fish. It's his natural lifespan that is very short, and that helps us study it in the short amount of time that we are trying to look into stuff. So if you don't mind, I'm gonna start sharing my screen and maybe start with a few slides, just so everybody here who's maybe not from a research perspective also gets a nice introduction of what we do and how everything works. So I hope you all can see my screen and the presentation. And as I mentioned, today I want to talk about the African Turquoise Killifish and how this novel model helps us to study aging, and especially for me, since I'm interested in vascular aging. So first off, why do we need to study aging? As you all have heard, what is commonly known as the age pyramid is currently turning more into a rectangle shape. So the problem is we get more and more people who are able to reach a higher age. But at the same time, the healthy lifespan these people experience don't grow at the same rate. Actually, a lot of diseases are primarily age-related. So, for example, cardiovascular diseases like heart disease, and of course, for me, very interesting, kidney disease. Things like neurodegenerative diseases like Alzheimer, for example, or just things like macular degeneration, which is one of the leading causes for blindness. And all of these diseases basically come down to aging. So a lot of researchers out there are doing excellent aging research, which is very awe-inspiring. But a lot of this research is being done in either non-vertebrate models, for example, in the roundworm, C. elegans, or in the fruit fly, Drosophila. On the other hand, there are vertebrate models like the mouse or the zebrafish that are being used. And if you look at this, you can see that a lot of these animal models are either being used here at the MDI BL or by our neighbors in the Jackson laboratory. So all of these models have benefits and they have negatives. So for example, in fast-aging animals like the roundworm or like Drosophila, they don't have certain aspects of aging like we have. They have no bones, they have no adaptive immune system, and most importantly, they don't have the vasculature. On the other hand, mouse or zebrafish doing aging research in those models that are typically more suited for developmental research takes very, very long time. So this is basically where the African Turquoise Killifish comes in. This is a model organism that we established here at the MDIBL, or was established around 2 years ago, and that currently 2 labs are using. So the good thing about the killifish, it's a vertebrate model. It has all the important organs that, especially for me, I'm very interested in researching. And it has this very short lifespan. So while we can let it age very easily, it's easy to generate large cohorts of aged animals that are very similar to each other. And so a little bit about this fish. It comes from Africa and it lives in those what you can see little pools of water that during the dry season completely dry out. So this fish has developed a method where it can spawn and reproduce very quickly. On the other hand, his short lifespan is basically just a trade-off for the ability to grow up and mature very, very quickly. So of course, as you can see, a fish that comes from the south of Africa to establish that up here in Maine has had some challenges. And this is, as you can see, just a schematic on what has to be done or how the fish is spawned and mated. And all of this plan or all those seasons the fish goes through in nature, we have to try to emulate in our lab when we do our research. And the nice thing, of course, is that this model closely resembles human aging, so we can use it as a close correlate, of course. And one of the things I think is very interesting is you can actually see
I think while Anastasia is trying to repair the connection, I mean, what she actually wanted to tell you, and you can see this, you can see that the fish age. You can see that they lose color, that they develop kyphosis, that they lose muscle. They are much thinner, and the color loss is quite significant.
Well, Dr. Haller, I know, this is we've talked a little bit about the fact that starting this new model is a bit challenging. And I think part of it is the work that Anastasia and others in your lab are doing is pretty new, right? This is the kind of, there's, how many other groups would you say in the United States or in the world that are using this model? Maybe talk a little bit about some of the reasons that you all are so excited about pursuing this work in the ATK model.
Yeah. Happy to do that. So Anastasia already mentioned that the model was introduced by two groups, and at the moment it has been very nicely characterized in the beginning, and then Anastasia is back.
Yes,
She is
I'm
back.
so sorry.
Okay.
So as
No,
you
that's
can
okay.
see, those are one of the joys of working in an older lab on the campus, but I'm surprised that happens too. So sorry.
Sorry about that.
At least the electricity is on, so this is good.
Wow.
So when
Sorry
did you guys
that we
lose?
lost you. Well,
I
let's
just
see. I
talked about
think you were just
the colors of the fish. That was all.
All right, so it wasn't that far. I just wanted to mention that, yeah, the aging hallmarks of the fish, and then I was mentioning that we are one of the very few people who actually try to research kidney and vascular biology in this new model system. So I wanted to ask if anybody has any questions up to that point.
Anastasia. I don't know if we really have said how long this fish lives. What's its lifespan?
So its lifespan typically is from 4 to 6 months in the model that we are using here, which is the GRZ strain, which is one of the shorter lives. There are also a little bit longer lived strain that usually lives from 9 to 12 months. But we here have chosen the GRZ strain, and this is good because this is, as mentioned, the shortest lived ones, and it's like the model that was very first established. So we have basically all the resources we need. We have a very well annotated genome, we have proteome and transcriptomic banks where we can look up all of our genes of interests.
Fantastic. So we are not seeing your screen, just so you know that, in case you want to share it again.
Yeah. All right, so let's see. I hope that now it works.
Yes.
Wonderful. Can you all see the presentation?
Yes.
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 Wonderful. So continuing on to that, one of the main theories we have about aging is this so-called vascular theory of aging. Basically, what it says is that of all the possible causes for aging or all its drivers, the phenotype you get from aging, a lot of the diseases that come out with aging are being driven by vascular aging. So this is like a common trunk of a lot of diseases that we later on see. And this kind of correlates to what you can basically see and what you can measure. For example, hypertension is a disease that also comes with aging because of the increase of arterial stiffness, for example. But if we look at vascular, not only as just the big arteries or the big veins, but there's actually a very small meshwork, which is the microvasculature. And during the aging, the microvasculature becomes less dense, and we call this process microvascular rarefaction. So there have been some studies that have been trying to quantify this phenomenon and see if it can actually be reversed. So what was shown was that microvascular disease, once we reduce the amount basically of aging we see, all of the different organs in our body become healthier. And that also leads to less like osteoporosis. It leads to less loss of muscle. It leads to hopefully less tumor burden and so forth. So one of those ideas is that this is basically driven by one of the larger systems, a molecular pathway of VEGF. And this is basically what we are trying to research, is to see how does VEGF affect the microvasculature in this fish? And are there other ways by which we can influence this system? And basically our idea is, if aging is a forward process to less blood vessels, can we reverse this process? For example, by trying to give like an anti-aging drug. And the complex we are interested in researching is the so-called SGLT2 inhibitors. So this is a class of drugs that was basically a blockbuster in the past years. It's a drug that is being developed from apple tree bark. The grand molecule is being called fluorescein. And we use the molecule called dapagliflozin, which is already a very established medication in nephrology, in cardiology. And the studies in humans that have been done with this substance show that it has a very extensive health benefit, even though what is interesting, we don't exactly know where this benefit is coming from. So what this drug does is it inhibits the SGLT2 transporter, which is basically a transporter that is solely located in a certain part of the kidney, a so-called proximal tubule. But even though we just reduce the activity of this one transporter, we can see all of this pleiotropic effects on the heart, on the liver, on the fat mass, and also on the lifespan of all of our patients. This was originally designed as an anti-diabetic drug. So it's basically being used for type 2 diabetes. And in the past years, we have been using it more and more, primarily in kidney insufficiency and for heart failure. And once again, it's very interesting for us, how is this process regulated, or how is this developed? So one of our hypotheses is that this drug may intervene with the microvascular rarefaction with aging, and this is something we are trying to research here. So, of course, all of this research cannot be done with a big lab, and thankfully here I have the opportunity to do this research in the Haller lab, and I'm very grateful for everybody who is helping us perform this research and gather our data. And I would be very happy to answer all of your questions and maybe get into some more detail.
Anastasia, thank you so much. That was a great overview of the work. I'm sure our audience has a number of questions, but I was really struck by this comment that you made about these SGLT2 inhibitors being found in apple tree bark. That's really fascinating. Do you have any more information on how that came about? That's really interesting.
Yeah, so the mother substance, which is fluorescein, has been known for many, many years already it was used as a drug early in the 80s. And after that, I think in the early 90s, it was developed as the drug group of the glyphosins, which is like the common name of all these drugs that we are using right now. And early on, there have been studies that were first trying to show its anti-diabetic effect. And the interesting thing is that nowadays, it's just not good enough for a drug to lower your blood sugar. I mean, there's many drugs who can do this. And in the past, there have been drugs that did lower your blood sugar, but that had severe side effects in humans. So nowadays, if you wanna like pursue your drug interest, you have to show that your drug is safe in a cardiovascular setting. So a lot of these studies on the glyphosins showed that not only were they safe, but they were actually able to like continuously have a beneficial effect on the people that were taking it. They had less albuminuria, which is like a sign that your kidney is basically leaky and not working well. They had a very well stabilized kidney functions over many years, which hasn't been shown for a very long time in other medication. And then, of course, this was for the field of nephrology. And then, of course, cardiologists discovered that it's a very good drug that helps additionally for heart failure to all of the drugs that we are using up until now. So it's not only like, let's say, wonder drug that helps us help our patients, but it also helps in the context with all of the other drugs and medications that we have been using. And I think that's very exciting that we're like progressing research and we are able to find more and more mechanisms that all help to heal our patients.
Yes, that's fascinating. So there are a couple of questions in the chat, and I would just remind everyone, if you have a question, you can feel free to use the raise hand feature in Zoom. You can type it in the chat, or you can just take yourself off mute if you'd like to jump in and ask. But Anastasia, one question in the chat is really asking more about the fish and sort of how do you actually obtain these fish from Africa?
Yeah, so the first GRZ strand basically was collected in the early 70s, 80s, and there have been several collections. So there are some universities that go out and collect every year basically the wild bred strand that they're using. But our fish are usually developed by other either research institutes or we order them. And they basically, this is the nice thing about this fish. We don't ship the fish, but because the killifish has this mechanism, we call it the diapause. So the fish can lay its egg, and the egg has certain mechanisms in place that protect it from drying out. Actually, a lot of those mechanisms were studied earlier in the bio lab here in the Atlantic killifish that basically can be fished just outside of my lab. And basically those desiccation mechanisms protect the embryo from drying out. And the African Turquoise killifish embryo is able to survive up from several months to several years even, being completely out of the water, being in a dry phase. And without it actually harming the longevity or the lifespan of the actual fish. So it's like some additional months or years that you can give to this fish. And we basically transport it into our lab in this dry stage. And once it arrives here, we can basically just add some water and then we have a fish. So on that, maybe a little anecdote. In the early 70s, the company that later developed the frisbee and the sea monkeys, they actually started out the company by trying to make instant fish. So like you have the eggs and then you put them in water and then you get fish. So they were trying to market that for children. And it was like a multi-million-dollar industry. And then they discovered something we discovered here in the lab too. It doesn't work. It's not, unfortunately, not as easy as it sounds. It's we're sometimes here surprised ourselves how this fish survives in the wild without us taking care of it. But it works. So it takes, like I mentioned, a lot of time to get them actually to the point where we can use them as a really good model for research, because we, of course, need certain numbers for that.
That's great. Dr. Boyer, you have a question? Nope, I think you're muted.
Can you hear me now?
Yes.
Yes. You also had the liver listed on your diagram, and I'm wondering what the effects of this drug are on the liver. Is it fatty liver disease that's primarily the focus there? Mm-hmm.
Yes. So what we think this drug does is something that's similar, for example, for something that's been shown for metformin. It's a drug that basically stimulates the fasting state. So you can think of it as fasting in pill form. So of course, if you have diseases that are associated with a turn on of metabolic activity, be it diabetes 2, be it non-alcoholic fatty liver disease, all those diseases that usually would benefit from fasting could benefit from the SGLT2 inhibition as well. The theories behind that, of course, is being that this drug might change how the nutrient sensing of our body is affected. It basically switches the metabolism in our cells from using glucose to hopefully being able to use more ketone bodies. So this is one of the ideas we are also trying to pursue and see if this drug is also similar to what we have seen in dietary restriction, for example.
Thank you. Very interesting.
Fascinating. So Anastasia, we do have a question in the chat about the name of the drug and whether or not you've seen any effects on cognitive function.
Mm-hmm. So the funny thing with the fish is they do show signs of neurodegeneration. There are some behavioral tests you can do on this fish to show that they actually experience memory loss over time. We have started establishing the model of our drug very recently, so to say. So the fish I have now are, I would say, like in their human years, in their 50s, 60s. And it will be very interesting to see how the behavior of those fish is like once they hit the actual old lifespan that we are interested in. And for the effect of cognitive function in this drug, I believe there's even some preliminary data in humans that have been able to show that this drug actually might have an effect. So once again, we don't know exactly what the mechanism of that might be.
Interesting. So one more question that we have here is, how might interstitial fluid and the newly discovered interstitial vessels that carry the fluid impact vascular aging?
So I think what Steve means with the question is basically what is between the vasculature, or like, let's say, between all the different kinds of cells, which, as we sometimes call it, is like the third space. So you have what is inside the cells, and you have what the liquid inside your blood vessels. And then there's like this imaginary, basically, third space where you have the extra interstitial fluids. So one of the ideas is that the blood vessels, they're supposed to be leaky only until a certain point. So you want like a diffusion of molecules like oxygen, and of course you want to take away all the CO2 which has been made up by the muscle, for example. And of course, we know that this basic homeostasis of the vasculature can be affected by VEGF. So VEGF was one of those molecules I showed you earlier, which basically stands for vascular endothelial growth factor. And one basically of the side effects this drug, this molecule has, is not only does it help little arteries to form and to grow, but it makes them more penetrable to fluids. So like if one of our ideas is how this drug might affect the system, we're of course hoping that it might make the vasculature more stable, so hopefully less leaky. But that for now is more of a hypothesis, and we have to see if our ideas work out as we think.
Fascinating. Any other questions for Anastasia from the audience? Anastasia, one thing I am curious about, I know you've talked a little bit about how your experience as a physician and treating patients really kind of drew you to the research side, this idea of personalized medicine. Can you talk a little bit about why you see that as really important in the work that you're doing in the clinic, and how those two things connect for you here?
So for me, for example, it was very interesting. I studied my work as a nephrologist, as a fellow in 2017, with my department chief being Professor Haller. And of course, all of my very young career, I always saw that medical doctors performing, be it clinical research or be it more basic research. And I was always fascinated by the fact that you can pursue as a doctor your own questions, and you can pursue those questions basically in interest of your work and in interest of your patients. Because we have a lot of patients that when we talk to, one of the things they ask us, well, why me? Why does this happen to me? Or for example, if you see patients on dialysis and you see somebody ages 10 years and 6 months and somebody else doesn't, you of course ask yourself, well, why is it? Why is this mechanism working that way? And I found it very fascinating to be able and to learn how to investigate those basically myself. And I think it also helps us to drive basically the knowledge, because of course I have more experience seeing how we work here with other PhD, with basic researchers, what actually impacts, let's say, our patients' lives. So I always find it very, very interesting doing the interdisciplinary work that we can do here. And I know our colleagues, or my colleagues, enjoy it too. And for example, just how to read a paper is so different from somebody with a, let's say, basic research background to how I as a medical doctor would view it. So we always have very interesting conversations about that.
That's great. And Dr. Haller, I'll throw this over to you, because I know this is a traditional strength of the MDI Biological Laboratory, this idea of bringing basic researchers together with clinicians, with physicians. And I'm just curious, as someone who has benefited, I think, from that, or certainly followed that model throughout your career, do you see that as something that will continue? I mean, is that a continued need going forward, just in general? Is there a specific role that you see MDI-BL playing in that arena?
Well, obviously, we have been doing this, and we have examples here, liver doctors who have been doing basic research at MDI-BL and taking care of patients all their lives. To be honest, I think that the best academic physicians are the ones who are doing basic research and are taking care of patients. As Anastasia just mentioned, one without the other is only half of the picture. So yes, I think that MDI BL has been doing this and will keep doing this. We all know it's challenging, not only when you come from Germany, but also in the US. However, I think we are becoming better. Anastasia has joined the lab at a moment when she's not only working together with researchers at MDI BL, but also at the Jackson Laboratory. So, Anastasia, how many young people are sitting around in our kidney meetings now from the TACs, from MDI-BL? These are numbers and an enthusiasm which we haven't had in years.
Yeah, I think so too. It's like many, I would say we're like 30, 40 people right now. And every time we meet, the consortium grows. And I think it's very interesting to like also set up all the collaborations that are possible here. So like the necessity of being able to see what is required in the clinic and how to translate that basically back from the bedside to the bench, I think is very interesting, as the same way going back around. So I think all those questions really help us to establish, hopefully, the MDI in the future as well, and to collaborate a lot more.
And just to add, we are role models. I mean, I can see this every summer. I can see this with our graduate students. I mean, we are models not only for a PhD career or working, doing basic research, but also being a medical doctor at the same time, so that you're able to talk about patients and to talk about both worlds. There are disadvantages, obviously. I mean, when you are a real PhD, you are more experienced, you're better with methods. So there is a payoff. But I've been sitting on the fence all my life, and I've been enjoying that.
Anastasia, can I ask you another question about the fish? You
Yes.
showed the atrophy of the vasculature. I don't know whether that was from the fish or not. Can you make such a measurement in the fish?
Yes, that's what we're trying to establish right now. So the measurement usually from the study I have shown you was performed in mice. The nice thing about a model like the mouse that has been established for many, many decades is, of course, a lot of research is tailored towards those, let's say, the typical or the canonical research models. So one thing, for example, you have antibodies for every protein in mice. It's different in the killifish. So for the killifish, I'm sure you have some experience in that. For right now, it's a very new model. It has been around maybe more on vogue in the past 5 years, which is a very short time in research. So, and seeing how nobody before us actually tried to research those type of questions, we're setting up basically how to investigate our questions. Because right now we are basically the first ones that have to look into endothelial cells of ATK. There's like 1,000 ways to look in endothelial cells in all the other mechanisms. But to see this in our fish is right now one of the things that I'm working on.
Can you use the new microscope to look at the vasculature?
Of course. So as some of you may know, we have an amazing microscope facility here at the MDI BL. And one of the bigger microscopes, or one of the newer, fancier microscopes, is, of course, the light sheet microscopy that we have here. So for me right now, before using the light sheet microscopy and setting up all the methods, we also have microscopes that allow me basically to make very fast, high put-through trials of the antibodies that we are trying to do. And I'm able to generate basically old fish very quickly. I can stain those with the various particles that we are looking at, and then we can image them and quantify basically within 1 day. So this is, I think, a very nice nice things that I'm currently spending my time on doing. Yeah.
That's great. Anastasia, as you kind of look to the future and your career as a physician, do you think you'll continue to do basic research? Do you think you'll continue to study ATK, or will you maybe move into another model?
Yeah. So I'm of course very enthusiastic about my model. So for me, you have to think a little bit. I think it's very difficult in the field of medicine and probably in the field of research to find, let's call it a small niche, like how our cells also look for a niche. I think it's very important to find a place where you love your work, you're interested, and you can maintain it for a long time. So I know that, of course, working in a new animal model for others may present huge burdens. And of course, it's very difficult to establish. But as Professor Haller mentioned, I'm kind of settled. So all I have is effort and time. So that is good. And I'm very motivated to be one of the driving factors, hopefully, in this animal model and especially in the field I'm interested in. I had the great opportunity last year to visit conferences on, of course, on nephrology and on vascular biology and aging. And the African Turquoise Killifish surprisingly hasn't really found its way into vascular research yet. And I would be very happy to be one of the first people to introduce this model there.
Yeah.
And as you can see, I'm also very ambitious. So that helps too.
And we are, I mean, Anastasia has started this work. And before we move on, I'd like to mention Eric Rogers, who started the research, and then also with the support of the Morris family, because when we started the killifish, this was tried. We needed extra support for that, and this was provided by Wissar and Martha. But thinking about the future, I know that Anastasia is working now for the next years at MDI-BL, but we are more than happy to have her back as a visiting scientist in 10 or 15 years and to support her work on vascular biology in killifish for the foreseeable future.
That's the price of admission, Anastasia. You never get to leave MDI-BL once
Oh, no,
you're here.
that's okay. It's like I will send all my grad students here, so they will want to apply.
If I may, I know this is a little bit private, but Anastasia, I think it's important for our listeners. You're living on campus. I mean, you're one of the people changing our campus. So you're living in one of the cottages with your husband and the dog. So you can actually see Anastasia in the morning walking the dog very early on. Different campus life than we had before in January.
Yes, I'm very, very grateful for that. So like this opportunity that I can come here and basically I arrived, I started doing the work and not having to look for an apartment here on the island or anywhere close, I think is a huge help. And of course, for me, it's also very good to have, of course, my family with me as a supportive system. Because as Professor Haller mentioned, coming from a well-established, I'm a Finnish doctor in Germany here and starting new wasn't always very easy. And of course, having somebody with me who helps me and a little bit of work-life balance so I can spend maybe just 10, 12 hours in the lab also helps.
Well, Anastasia, it's been such a delight to hear a little bit about the work that you're doing. We're so excited to see where this work leads. I hope you'll come back and give us some updates on how it's going. I know I know it's been a challenge, but also I hope very rewarding. And we appreciate your spending some time with us this evening. And it's fun for us to be a little bit part of your journey. And again, thank you so much for sharing your work with us. So thanks to all of you as well. Dr. Haller mentioned, the importance of funding in keeping research programs like Anastasia's up and running. Certainly building out our graduate and professional education programs has been a huge priority for us over the last year. And I think you can see tonight why that is so important. And so we're grateful to all of you who have supported those endeavors, and look forward to sharing more about more of the stories of these outstanding young people who are now here and a vibrant part of the MDI BL research enterprise. So thank you so much for that. Just a quick reminder that our next Science Cafe will be February 12th, and that will feature another MDI BL scientist, Dr. Helena Shabata. She's relatively new to the laboratory, and so we encourage you to tune in, learn a bit more about her work. She is really looking at how the genome is regulated and how that sometimes when things go awry, how that leads to specific diseases, things like dyslexia, things like diabetes, all these muscular, very rare disorders, but just a wonderful opportunity to learn a bit more about how these model organisms, she is using the fruit fly, so another, important organism in research. And so we invite you to tune in with us. We'll have another wonderful conversation. And again, our thanks to all of you for your support of these science cafes. It's a wonderful way to engage in conversations about the science that really impacts our lives. So thanks you all so much, and we look forward to seeing you next month.
Thank you.
Thank you.
Thank you very much.
Bye-bye.
That was very fun. Thanks for
Thank
having
you.
me.
Thank you.